And therefore, as a free man, I take pride in the words, Ich bin ein
Science! Science!
I know the human being and
Science!
can coexist peacefully.
This was thou findest.
Guys, this is Petri Dish. We're on our part 2 of our crazy CRISPRs and gene modification episode. I'm here with Sean David Allen, and I'm here with Andrei Day.
I'm back.
He's back, baby. Woo. He didn't leave the room between recording these 2 episodes. And so we've talked about all that nerdy shit about what CRISPRs really is, but now we gotta get to that good gooey goo stuff. We gotta talk about what can you do with CRISPRs? What can you do with modern gene modification? How is it going to destroy the world?
All right, everybody calm down. Fucking pessimist.
That's what I came here to talk about.
All right. So, what have we done with CRISPR? Let's get into it.
So, gene modification is one of those things that in sci-fi we always talk about humans, but we've already dramatically gene modified certain things out there, right?
Yeah, and you know, we've already tried our hand for thousands and thousands of years with selective breeding, and now with these tools we can just kind of, in a much more targeted way, try to affect plants and animals, and we've already done that even with CRISPR in some cases. So I took some notes. I read the literature. I took some notes.
You went to the farmer's market and listened to people complain?
Oh, yeah.
Because we are, trigger warning, we're talking about genetically modified organisms.
Oh, yeah.
Which, extra trigger warning, you dumb if you don't eat them.
No, that's not fair. There's lots of kinds of GMOs out there, and I think...
They're all delicious.
If you talk to scientists that make GMOs, and you said, I don't like Monsanto, I think you'd find a lot of them don't like Monsanto, too. So I don't think that's like, anyway.
Don't offend our main sponsor.
You... Uh-oh.
Oh, I shouldn't have said it. Oh no. Oh, no.
All right. So, let's start with some plants. Okay, so for example, strawberries have been CRISPR'd. And strawberries are kind of an interesting example because they're a type of plant that's similar to a lot of other plants that we eat in that they have multiple copies of their genes, way more than just 2. So in humans, we're what's called diploid. You get 1 copy of all of your genes from your mom, and you get 1 copy from your dad. Strawberries, unlike humans, we have 2 copies of the genes, 1 from mom, 1 from dad. Strawberries have 8 copies of their genes. So
What the fuck?
They're called octoploid.
What?
And so what that means is that if you want to change a gene, like even if you want to break a gene, unlike in people, you need to break 2 copies. In a strawberry, you need to break 8. So it's much harder to do genetic modifications to those kinds of plants.
Why do we want to fuck with strawberries anyway?
Maybe you want them to not be quite so red anymore. Maybe the redness pisses you off. It gets you hot.
I want purple berries, dude.
Yeah, maybe a little bit of that or something. Or maybe we want to make strawberries that are bigger or pest resistant. No more, strawberries nibbled
More
on.
shelf stable.
Yeah, sure.
Able to be transported further distance.
Okay, guys, I get it. There's good reasons. All right. How do you do it? How do you oploid these octagons?
Holy shit. What? What did you just say to me?
Come on, don't let me slow you down, Sean. Come on.
We entering the octagon? Okay. Yeah, so while I didn't put this down in the notes, I've actually looked into how you get CRISPR into plants. And there's 2 ways. One of them is you use something called agrobacterium. And so what it is we have a lot of ways to get genes into bacteria. That's relatively easy for us to do. And there just happens to be a bacteria that has evolved to put its genome into plants. So you can grow this bacteria, put CRISPR into the bacteria, and then smear it on plants. And then the bacteria will get into the cells and put that CRISPR DNA into the cells for you.
So what's going to stop, I remember Andre saying this, blowing all his good sauce in the commercial break.
Yep.
But what would stop like a terrorist getting some of this schmear and just throwing it on a strawberry, and then suddenly, if you eat a strawberry, you like fucking turn into a terrorist?
Wow, that is a serious
Left
departure
turn,
from
baby.
what I was talking
Turned
about.
left at Albuquerque.
We're going to talk about the terrorist gene later in this episode.
I'm just quoting Andre. Andre said all that.
But-
what an idiot.
I don't know, like $20,000? I think that's what's stopping them.
That's
That's not a lot of money.
I feel like a Wahhabist has that.
All right. Well, we'll get into some of the ethical implications of some of this CRISPR technology, but main point is you can do the agrobacterium route, or there's another way to do it. You can take the DNA, like the CRISPR DNA, put it onto gold nanoparticles, and then shoot it into the plant using something called a gene gun.
Dude, that's so cool. Wasn't that didn't Arnold Schwarzenegger have one in Total Recall?
A gene gun.
This is my gene gun.
Is that how he made the creepy stomach alien
Yeah,
thing?
start the
Are
reactor.
these gene guns also protected by our Fourth Amendment? Or Second Amendment?
Yeah, I was like,
You
which one's the-
liberal cuck.
Can't even count no good. All right. Gene gun is actually a very cool technology. You take the gold nanoparticles that you've attached the DNA onto, you load it into these little tiny chambers where you dry it, and then you use basically helium, highly pressurized helium, to just blast the gold through the cell wall of the plant into the cells. And then the CRISPR then takes off and does its job.
So why can't we do that with like human biohackers?
Oh, yeah, shoot them with gene guns?
Yeah.
So one thing I've looked up is
Needles
whether or
are
not
cheaper?
That's probably true.
plants don't have as many rights or feelings as human beings do.
I think biohackers kind of write those off when they've decided to call themselves biohackers.
I'll say this much. I read one paper from 2009 where they developed a low pressure gene gun that's supposed to work through skin, which I think could be really cool for maybe someday making tattoos that like glow a color or something like that. You know, like
Don't they already have those?
yeah, but that's with ink. I'm talking about with your cells intrinsically have them make protein that glows. So we've done strawberries, and what we did with strawberries was just a proof of concept to show that we can do CRISPR. All that we did was we broke the gene that makes them red, so that we just had a bunch of white strawberries.
Weird.
Ooh, albino strawberries.
Yeah, it was just a proof of concept.
I would probably sell, though.
Yeah, it could be. Some people might like that
You
thing.
could sell any sort of stupid shit in Japan, though.
Japan, I was thinking like, some Edelweiss shit. You know what I mean?
Yeah,
Like,
some Ku Klux Klan.
yeah.
Crackerberries.
But anyway, we've also done grapes. Okay, and grapes are similarly a high-ploidy plant that are kind of difficult to genetically modify. And in this case, what they did was they introduced a gene that makes them resistant to a fungus. So that means that, French fried potatoes and Californians can enjoy their wine more because it won't get eaten up by fungi.
That's good. Unless it's on fire.
Yeah, I mean, we're all drinking,
Other problems.
we're drinking wine right now, so I think we can all appreciate how important that is.
Maybe we can bring back the Michael Gross banana.
So,
I see we got wheat here.
yeah. So this one group went in with CRISPR and broke the gluten genes.
Why the fuck would you do that, dude? Gluten is what makes pizza good.
Right.
Which Andre was supposed to bring pizza, though. I meet him, and he's like, no. I was like, what the fuck?
Yeah, so I'm not entirely sure what the point of wheat is once you get rid of the gluten, but they did it successfully, and that's impressive because not only is there, I think wheat might be hexaploid or something like that, but there's more than 6 copies of the gluten gene in wheat. It's been duplicated several times, so there's a bunch of spots you had to hit. It's very hard to genetically modify.
Mesopotamian.
I'm sure it's even more complicated than that because, like, gluten is actually formed by, like, a conglomeration of different proteins, like gliadin and stuff like that.
Right.
Like, they combine to form gluten.
Right.
Mesopotamians loved gluten. And
You're the historian, so I can't dispute it. I don't know. I
I'm
don't
a
know.
bartender. Okay, so I like how you have tomato here and then no notes, just tomato.
Yeah, so tomato we've done a lot of different modifications for, and
That's why they're so bland and tasteless these days.
that's very true that there was a tomato, now ironically named the Flavor Saver tomato, that was a genetically modified tomato, and what it was modified to do was to be more shelf stable. So using technology, this was before CRISPR, they broke some of the genes that normally cause the tomato to decay as quickly. What they didn't know is that they also accidentally broke a couple of the flavor genes. So those tomatoes taste like something ranging from nothing to sewage, which is why like heirloom tomatoes are so much tastier and everything like that.
Scientists are gone out of control.
But now with CRISPR, there's dudes going in, they've learned from their mistakes, and they're trying to make more shelf-stable tomatoes that also don't taste like shit.
So even better tasting Franken tomatoes.
Yeah, exactly.
I like
Beautiful.
it.
Beautiful.
I see rice here.
Yep.
Rice, as you know, we're Oriental, so that's really close to our heart. So tell me how we've ruined rice with genetic modification.
I guess you can say Oriental.
Yeah, I can.
Yeah. You're
People
like-
don't know I'm half Korean. Oh, shit, I said it.
So one of the things that they've done is they've engineered rice to be resistant to a kind of virus.
I see Tungro.
Yeah, rice tungrospherical virus. Which
Tungro.
is a cool name. But they've also done another experiment that I think is really interesting. There's a lot of varieties of rice out there, right? And some of those varieties of rice are ones that have had natural qualities that we've bred into some strains because they'll do things like be drought resistant, or they'll be resistant to fungi, or they can be grown at higher temperatures. So there's all these qualities to rice, or maybe they're more nutritious, right? But one of the things that we found is that there's this kind of spectrum in rice of how much amylose content they have. And the more amylose they have, the less kind of plump and tasty they are. You basically go from sticky rice on one end, which is very low amylose, like, what is it, glutenaceous? Glutinous, yeah. Glutinous rice. So glutinous rice is all the way on one end, low amylose. And then you have rice that's like way past basmati. Something that's like, it's kind of tasty once you cook it, but then it gets dry really quickly, right?
And that's high? Amylose?
That's high amylose. And basically they found out, hey, we can break a protein. And that protein normally takes amylopectin and turns it into amylose. If we break that, we can turn something like basmati rice into a sticky rice.
Why wouldn't you just use sticky rice?
Because the sticky rice might not have had things like drought resistance bred in.
Oh.
And in some cases,
They're very spoiled with their water in East Asia.
well, I mean, not to jump too far back, but in the climate change episode, we mentioned that rice paddies generate a lot of methane. And part of what they think they can do there to reduce methane production is to have it flooded less of the time. But if you do that, you want rice that's hardy enough to be able to handle those conditions.
Sure, because usually rice you just like have growing like in water most of the time, don't you?
It doesn't need to, though. The reason they do that is actually weed abatement.
Right.
Lots of plants can't grow in that high of moisture, but rice can, and that's why they flood them.
So, yes, that is exactly what's going on. And that's actually the decaying weeds is actually one of the main... the bacteria eating those is what causes the methane and the nitrous oxide production.
Man, we don't say it enough, but science is so cool.
So, there might be wild rice varieties that, for example, are more resistant to weed invasion. And Well, hold on. Don't jump to rapeseed.
What's rapeseed?
I got one more.
Okay,
rice
shit,
thing.
I'm sorry.
We've also incorporated bacterial toxin genes into rice. But in this case, they're bacterial toxin genes that are not toxic to us. They're toxic to insects. And what we've done is we've actually taken two toxins, fused them together, and put them into rice to basically make it so that insects have a harder time eating the rice. So it's a pest resistance mechanism.
So what's stopping a sufficiently motivated asshole from having rice produce a toxin that is toxic to us and just
Like Joe
scattering
Biden.
that in the wind?
What's stopping Joe Biden from doing that? Okay,
Not
Silverback
who I
Joe.
had in mind specifically.
I mean, the answer was litriteracy. It's hard to read when you're that old. Just kidding.
Poor Joe.
I love Joe. Joe 2020.
Wow, is that it?
I'm just hopping between extremes.
All right. What's stopping somebody? Mostly just money.
But it's getting cheaper all the time.
Yeah, realistically, the money amount to do something to rice is not that expensive. It'd be like, in the hundreds of dollars. The question is verifying that you've successfully done it, which will take time because you'll fail a lot of times. That's just how biology goes. I don't know if...
Can't you just throw the rice at like and just see what happens?
Why are you so racist?
No, well, I'm just saying.
Pick somebody else.
Well, if you're the kind of person who's trying to create poisoned rice, you're going to probably do it in because there's less regulatory mechanisms to catch you, right? So you probably get a or you and you throw rice at them and or have them eat it. I don't know, I'm not a scientist. And then if they live or die, you've just validated or verified it, right?
Pick someone else to throw the rice at.
Okay,
I'm tired of you targeting the...
See, Andrei agrees with the basic premise of what I'm saying if he doesn't agree with the nouns. Tell me the answer.
There's not that much stopping somebody. No, I guess, so from the scientific perspective, all of that science is eminently possible. In all of these cases where we've successfully put in a gene to do something good, you could have picked a gene that would do something bad and had pretty much the same success rate.
So we should just ban this entirely.
Yeah.
Good luck.
Okay, so I see here rapeseed
Yep.
and reishi.
Yeah, reishi mushrooms. Yeah, sure. So rapeseed oil is canola oil, right? And one of the things is when you're harvesting rapeseed, sometimes the seeds will just break upon harvesting. So you lose a lot of oil just during the harvesting process. They can just break apart and your machinery will have just a bunch of canola oil on it, basically. So what they've done is they've been able to CRISPR rapeseed so that it is less likely to break apart on harvesting, but still able to break apart when you're harvesting oil out of it. So it can increase yields.
Okay. And then how about these mushrooms?
Yeah, so reishi mushrooms are cool because they're like, they're a fancy mushroom guy that is used in Chinese herbal medicine.
Medicinal shrooms.
It's pretty popular now in like the goop crowd. You can make a tea out of it that's supposed to be good for your gut and your health. And hypothetically, reishi mushrooms might have it wouldn't be the first time that mushrooms have molecules in them that are bioactive, right? That shit really, that
I
happens.
can tell you that from experience.
Yeah, exactly. Which, someday soon, I've written up the notes for us to have an episode on psychedelics. So we'll do that eventually. But in any case, we have done a proof of concept in reishi mushrooms where, again, what we were doing was we were breaking a gene for a certain kind of pigment. So we were making like albino reishi mushrooms. It didn't do anything to the nutritional quality. It's just thus far, we've had almost no success in genetic modification of what are called higher fungi, or, like not yeast, basically. Yeast, we do genetic modification on all the time, but higher ones like mushrooms, they've been very difficult to do with a lot of the tools that we've had. But CRISPR, you can mess around with it so much, there's so many different forms, and you can do it so cheaply that we figured out combinations that are successful. So, reishi mushrooms are the first ones, but you can imagine CRISPRing a lot of different kinds of mushrooms.
Can we get to a point where we could take the LSD proteins and then put them into, like, chicken? Well, what the fuck are we talking about right now?
So
like you eat a piece of chicken breast and then you're tripping for like 12 hours?
In a perfect world, yeah.
Yes. So you're presaging a section down the road where I talk about some of the future stuff. But yes, I think that's possible. Now, technically, LSD is a synthetic product based off of ergotamine from ergot, which is a kind of fungus that grows on rye. So there's a bunch of enzymes for making the ergot chemical, and then there's some synthetic stuff that we do, and so we need to figure out enzymes to do that. But maybe someday. Or what about THC? THC chicken.
Okay, well, now that we've jumped the shark talking about...
KFC, man. I'm getting my chicken at THC chicken.
That's the next Popeyes thing.
So now that we've successfully talked about drugs, let's take a break. After that, we're going to talk about some of the really crazy shit we can do with animals, and then we're going to get into all the evil shit we can do with humans.
Sean here. As you guys know, I'm a great scientist and a loyal lover. But I am ugly, and because of that, my beautiful friends periodically spit on me. Studies prove that good-looking folks are black holes of empathy, incapable of the basic acts of kindness that make civilization possible. If only they could get beaten with an ugly stick and finally understand the world as we ugly people live it. Now there's the perfect service for your beautiful friends, Ugly Stick. You sign up for the service, and empathy artists will arrive at your friends' apartments or homes, extreme makeover style. They uglify your friends so that they can internalize all of the things we uggos go through every day. Your friend will become a more conscientious person, beautiful inside and out, thanks to you and to Ugly Stick. So sign up today. Make the world a more beautiful place by making your friends uglier.
Okay, guys, we're back with Petri Dish. We've talked a lot about all the different ways that Monsanto is genetically modifying your food to be better for you. Shut up, hippie. Now we're going to talk about animals. What are all the wacky ways we're going to fuck with God's creation? Sean, I see in the notes here just silkworms proof of concept. I don't know what that means. What are we doing with silkworms?
Yeah, so I think the rationale behind people trying to genetically modify silkworms is that they're what we get silk out of, but like it's kind of silly to just have silkworms make one cocoon for themselves and then steal that and try to make clothes out of it.
Super rude.
Whereas, maybe we could genetically modify them to make a shitload of silk and never use it, right? So we might be able to speed up the process. But anytime that you want to do anything like that, you usually do an experiment first to see whether or not that animal, you even can figure out a way to do CRISPR on them. And in this case, they have successfully done CRISPR on silkworms. So I imagine this lab, their next step will be to start modifying different genes.
What was the change they made to silkworms?
Yeah. So a really, really popular proof of concept step for most of these genetic modifications is to break a gene that makes a pigment. Because then you can just look at them and you can tell like, what percentage they worked. Right?
This thing is a different color.
Right. Instead of having to sequence their DNA. Right. That's exactly what it is. It's a huge cost saver to just have a proxy like that. So in a lot of these cases, the proof of concept is going to be just breaking a color gene.
Can't you just open its butt wider and then like more silk would come out at a time?
Do silkworms have like butts?
I mean, the silk's gonna come out of something, right?
I don't think they poop the silk out.
Okay, Mr. Smart Guys. I don't know if it's anatomically called a butt, but it's
they
like
do
a
have
silk
butts.
asshole.
They do have butts. God damn it.
I mean, they poop, but like so do birds. They don't
You're
have
such
butts.
like a fucking proctologist. You're always trying to get up inside that butt.
I had a terrible childhood.
What? We had the same childhood.
I am what you did to me.
Whoa. We just did some shit. Wow.
On the
right
next
here.
episode of
You realize you just said that about me. I'm right here.
Don't worry, Andrei's here
All
to litigate this problem.
All right. We've also done pigs.
Yeah, what have we done with pigs?
With pigs, we've removed a series of retroviruses. So basically, retroviruses are a kind of virus where they put their RNA into your cells. That RNA gets turned into DNA, and then that DNA gets shoved into your genome somewhere. And for pigs, when they get infected with a retrovirus, they are left with these retrovirus sequences in their genome in places. And the danger is that maybe someday those retroviruses will stop kind of sleeping in the genome, will wake up, and that pig will be reinfected with that retrovirus. It's one of the reasons why like HIV or something like that can be latent for so long in people, is because it'll kind of incorporate itself into the genome. So for pigs, these retroviruses are called porcine endogenous retroviruses, and porcine endogenous retroviruses, or PERVs, are something that these scientists wanted to CRISPR to break so that they could never turn back into the retroviruses. The reason for doing this is they want to eventually use those pigs for xenotransplantation.
We want to like grow human organs inside the pig, slaughter
or at
the
least
pig.
put pig organs in people.
Yeah, so we want
gross.
to take pig organs, and we want to be able to, people who need a new kidney or need a new liver or a new heart valve, put those pig organs in, and hopefully make them as human as possible so that we don't reject that organ.
Is a person who's filled with pig organs still a person, or are they a pig now?
Is this like one of those
Ship of Theseus
kind
yeah.
of questions? Yeah. What percentage of a person can you replace with pig before they're just a pig?
Yeah.
I don't know, dude.
Of course, a lot of capitalists are already pigs.
That's for the philosophers to decide.
This is unrelated to CRISPR, but something we're already doing is like taking organs from animals, sort of rinsing off the like animal cells and then replacing them with human cells.
What the fuck?
Yeah, so it's a process called decellularization. And what the main point is that organs are made up of cells, but they're also made up of all of this shit that cells excrete out into a matrix. That's sort of like scaffolding that forms the shape of an organ. And you can take a heart and wash it with a detergent. And the detergent will basically wash away all the cells, but leave behind the scaffolding. And you can put in new cells.
Cells that are harvested specifically from the person who is like anticipating this donation of an organ.
Right. So we didn't talk about stem cells on their show yet, but you could take somebody's skin cells, turn them into stem cells, put them on these decellularized hearts from like a pig.
And then have those stem cells turned into heart cells.
Yes, and then you'd basically have
And they would be identical to the heart cells that already exist in that person. So there's like no chance of rejection.
Yeah, immunogenically identical. This whole process isn't like solved perfectly.
Right,
You know what I mean?
right. I
Especially
mean,
for
it's-
more complex organs. Hearts are actually relatively simple as far as organs go, but kind of more complicated ones like livers are a little bit harder to figure out. But, whoa.
Yeah, we're getting ahead of ourselves.
Decellularized organs, xenotransplantation, that's all shit that's in the notes for a completely different episode that I've already written. But whatever.
Okay, look, we got to get to humans, so let's checklist this. What's another animal that we can do cool genetic modification with?
Locusts. So with locusts, we've broken a gene that they use for detecting odors. And normally, so locusts form these huge swarms, right? And those huge swarms can be ecologically devastating.
Now, locusts are just grasshoppers that have sort of picked up on each other's like pheromones or something. And when they turn into locusts is just like when they go on like pure like rage mode and just like swarm and eat a bunch of crops, right? Or am I wrong?
Right. So I think that there's certain species of grasshoppers that have a higher propensity to do the swarming thing. But yeah, so when we're talking about locusts, we're talking about like in swarm mode. Mm-hmm. And the swarm mode is based, like you said, on pheromones. If you break the gene that they use to sense that pheromone, they're still alive, right? They still go around and do their grasshoppery business, but they can't swarm anymore. And so basically, we're not removing them from the food chain. We're making them so that they can't do the kind of extremely devastating swarm behavior that they can do.
But how do you replace the ones that do have this gene in nature with the ones who don't?
Right. So that's a good question. That kind of plays a little bit into what we've done with mosquitoes, which is something called a gene drive. So a gene drive is a kind of gene configuration that makes it outcompete the standard gene. So that means that over time, as it mates into the population, the modified gene will outcompete the standard one. Or in the case,
so that to me seems counterintuitive, because if you're breaking the pheromone detection of an animal, you'd think it wouldn't be able to procreate as well as one that could detect the pheromones.
Yeah, so luckily the pheromone behavior is mostly about feeding and a little bit less about mating, in that there's a really specific odor detection for the swarming and not for mating. So they're still capable of mating. But in this case, kind of what you're trying to do is you sort of create a new gene. So it's not about breaking the gene and passing on the broken gene. What you're passing on is CRISPR. You keep the CRISPR in the locust genome. And every time they mate, those kids have the CRISPR that re-breaks the gene over again. So even if the kid is what's called heterozygous, they got a functioning pheromone from their mom, but a broken one from their dad.
Oh, the CRISPR breaks the functioning one.
Exactly, right. So it turns them homozygous by breaking it in the kid. A similar kind of gene drive has actually been used in mosquitoes. In this case, the gene drive is supposed to make it so that the mosquitoes become either sterile or incapable of supporting
malaria.
Yeah, incapable of supporting malaria, the protist that causes malaria. And so, in that case, being able to have that gene drive there should support it through generations that can make it so that those mosquitoes continually can't be hosts of the malaria in that area.
That'll save a lot of lives.
Yeah, mosquitoes as a vector, the set of diseases that they cause are the one of the main leading causes of death in the world.
I kind of like Bill Gates' approach, where he just wants to put a bunch of robots out there that shoot lasers and then just shoot the mosquitoes out of the sky.
That sounds like a lot of fun.
Did he say that?
That's a real thing. He's funding.
I like it.
I'm glad he has money. But
I think he's funding a lot of stuff. You know what I mean?
Yeah, no, he's definitely like, that's just one of many things he's doing to try to eliminate malaria.
There's actually, this has nothing to do with CRISPR, so it's a scientific digression, but there's a bacteria called whaddya, and whaddya already exists in almost all flies in nature, but almost no mosquitoes in nature. It's a not very aggressive infection. It doesn't do that much to the organism, but mosquitoes infected with whaddya bacteria can't be infected by viruses for like Zika or dengue. And so basically they're taking a bunch of mosquitoes, infecting them with whaddya, and then putting those out into the environment so that they can kind of spread that to other mosquitoes. And that seems to be pretty effective so far. Anyway.
All right, so I see something that's really close to your heart, mice. right?
Yep.
I don't know if you guys know this, but Sean loves mice and keeps a lot of mice. So Sean, tell me about how we can work with mice.
Yeah, so we use mice a lot in a lot of our research studies. It's one of the places where if we want to know what a gene does, sometimes we'll break that gene in the mice. But to do that, you need to breed a strain of mice that has that gene broken. That's not very easy to do. It takes a lot of time. CRISPR can do that a lot faster for us. So on the research end, we can figure out the function of genes a lot faster. Another thing is that mice are not the same as people, right? So sometimes we'll do studies in mice and we'll like cure something in a mouse. And then we try it out in people in clinical trials and it doesn't fucking do anything. And the reason will be because mice are different from people. And maybe what we can do is use CRISPR to introduce some humanized things into the mice, make them a little bit more human-like.
The rats of Nim.
Yeah.
Redwall.
Yes. Mrs. Frisbee and the rats of Nim. Yeah.
In a way, they're more human than humans.
Oh, they're definitely more human than humans. They're already more human than humans.
So I'm just going to check off some animals and let me know if there's one of them you want to talk about really specifically. But we also got cows, goats, salmon, and all sorts of fucking monkeys. These are all sorts of animals that we have a very close relationship with that we're starting to use CRISPR's Cas9 in really interesting ways.
Yeah, so I mean, all I'll say on the goat one is that cashmere goats are really cute. And I think people should look up a picture of cashmere goats because they're adorable.
Sean's friends with one that he's named Sarah.
Yeah.
Yeah.
Sarah's a cutie. And
Stop it.
well, it's just the genetic modifications for the cashmere goats is trying to make it so that there's higher yield on the wool. So they can just grow more wool that we can use. And then you can have fewer of the goats, or you don't need to try to get the wool off them as frequently. They'll just grow more. Yeah, so I mean, I guess that's a lot of the animals. Maybe a non-comprehensive survey. I think we've done just like way more.
Right. There's all sorts of shit, really.
Yeah. Yeah.
I mean, just the other day I saw an avocado that was pink. You
What?
know? No, I didn't see that.
Oh, okay.
Anyway.
Was it just painted pink?
Yeah. It was just a worker at Trader Joe's, just kind of like dead in the eyes, just kind of painting things pink.
Yeah.
But okay, so all this fucking animal shit. Humans, man, this is where it's at. Tell me about some sexy stuff we've done to Chinese babies.
Yeah, okay. We can start with the Chinese babies. There's a really controversial thing that came out where I think a lot of people know about this. This dude scientist basically took a bunch of embryos and tried chopping or breaking the gene CCR5. And CCR5 is a protein that's on the surface of cells. It's the way that HIV gets into T cells. So he was basically trying to make it so that these babies can't get AIDS. Is nice if it worked. The thing about CRISPR that I've mentioned is that regular ass CRISPR-Cas9 can have off-target effects. It could cut somewhere else in the genome, right? And who knows where it's going to cut? Who knows if it's going to be in a gene that normally guards against cancer, and then now maybe these, girl twins are going to get cancer when they're 12 or something like that, right? So it was extremely potentially dangerous to these kids to do this without any kind of knowledge of how successful the on-target effect was.
Of course, they don't have Second Amendment rights in China.
Right. The embryos did not have access to guns, so they could not shoot the scientists before he CRISPR'd them. But so aside from this...
More embryos had guns, though.
The only thing that stops a scientist, a bad scientist with a gene gun, is a good fetus with a gun.
So, aside from this this experiment of really dubious ethics, there's been a lot of other uses of CRISPR and other genetic modification tools, the talons and the zinc finger nucleases, in humans already. And so there's actually clinical trials using CRISPR for people. We've done it for blindness. There's a genetic blindness called Leber congenital amaurosis 10. And out of all blind people, I think it's something like 15% of them are affected this way. And it's because they have another single nucleotide mutation that basically causes a certain protein in the eye to be way shorter than it's supposed to be. So it doesn't work as well. Okay? And so they become progressively more blind, and then they're just blind. And in this case, what the CRISPR is doing is it's cutting right nearby that messed up nucleotide and breaking the gene in that spot. And that's supposed to restore the full function of that gene. It's a little complicated because I don't know how much detail to go into on this one. The main point is that in humans, you have a gene, but not all of the gene codes for the protein. It's just chunks of it called exons. And in this case, the mutation is in an intron, a part that doesn't normally code. But because of that, just one mutation, it messes up the order of introns and exons, and so it just completely fucks up the protein. And so what this does is it breaks a part of the gene that doesn't code for a protein. But by breaking it, it allows for the actually correcting parts to come together in the correct order.
And I'm not just seeing, I'm seeing, like, so that's blindness, which is totally fucking nuts. But I'm also seeing sickle cell anemia.
Yeah.
I'm seeing this autoimmune deficiency disease, whatever. I'm seeing fucking Chinese twins disorder.
Well, we did the
yeah. I'm just, so there's like all sorts of diseases that we could actually we could cure with CRISPR-Cas9.
Yeah. So, sickle cell is a fun example because, what's going on in sickle cell is another single nucleotide mutation that basically breaks the adult hemoglobin. So you have a sort of misshapen blood cells, but part of it is they can't carry oxygen as well anymore. And in the process of becoming misshapen, they can also get kind of clogged up or break in your arteries and veins. What this cure is supposed to do is to break a protein responsible for turning off fetal hemoglobin. So fetuses have a completely different molecule in their red blood cells.
What?
Yeah, so that they have fetal hemoglobin. Normally that gets turned off when we're kids, and we switch over to the adult version. But in these people with sickle cell, the adult version's broken, so for CRISPR, we just turn back on the fetal one.
So wait, rather than like fix someone's red blood cells who has sickle cell anemia, we're like reverting to like a different red blood cell entirely that adults don't even usually have.
Yeah, more or less. A red blood cell's got a lot of components to it, and the hemoglobin is just one part, but we are switching to a fetal one instead of the broken adult one because fixing the adult one requires the lower probability HDR thing, right? Fixing it is harder, and in this case, they're not breaking the fetal one. They're breaking a protein that normally turns off the fetal one. So it's even one step further removed, right? But by doing that, and so what they're going to do is they're going to take the bone marrow out of these people. Bone marrow is where your red blood cells come from. Right. They're going to take those cells, treat them with CRISPR, pick out the ones that were treated appropriately, go back to the people, give them chemo to kill all of their bone marrow, because the bone marrow that they have is making shitty blood cells, and then reseed their bones with the CRISPR'd bone marrow.
Wow.
And if it works, it's in clinical trials. If it works, these people might be cured of sickle cell anemia and beta thalassemia. Thalassemia, yeah.
Okay, and then I see we got this autoimmune deficiency disease, SCIDX1. We treated 29 babies. 24 of them ended up having functional immune systems, and 5 of them we fucking gave cancer.
Yeah, so SCIDX1 is kind of like the bubble boy disease. It's basically a severe immunodeficiency where you can't protect yourself from infection. So there's a very considerable hit to quality of life.
You're just gonna fucking die at some point.
Yeah, or you'll live a not very fun life kind of locked in, right? In a situation where you can't really go outside.
If I was a bubble boy, I would take those odds.
Right, so these babies were treated. And in this case, this was not CRISPR-Cas9. This one was actually them just using a retrovirus. So they used something kind of like an HIV virus, except they filled it with a functional copy of this gene. And retroviruses integrate that DNA into your genome, but at random. You don't know where it's going to go in. So unlike CRISPR-Cas9 or zinc finger nucleases or TALENs, all of those guys are targeted to a spot to do something, right? Retroviruses, just putting a gene in a retrovirus will mean that gene gets put anywhere.
But certain retroviruses don't target certain cells. Like HIV targets like your white blood cells specifically, right?
Yes. So in this case, they used a gamma retrovirus that does have a preference for certain cells. And in general, retroviruses pretty much only work on dividing cells. But dividing cells are like the ones that make your immune system. And so that's why that was still useful in this situation. Because you want you're trying to fix the function of the immune system. It's an immunodeficiency. But the thing is, within those immune cells, you could have the functional gene go into a spot of your DNA that wasn't doing anything, or you could have it go in the middle of a gene that normally inhibits cancer, which is what happened in some of these kids. 5 of these kids ended up developing cancer because the retroviral therapy basically broke some of their genes, which is not great.
But if we had CRISPRs for this experiment, maybe it would have worked even better because you could be more targeted with it.
Right. So, in this case, 24 out of the 29 babies were fixed, but out of those, 5 ended up getting cancer, right? I think that with CRISPR-Cas9, you'd be less likely to get the cancer. And if you do one of these kind of fancy HMEJs, maybe you can also get 24 out of 29. That might not be as easy.
Hey, speaking of cancer,
Yeah.
could we use CRISPR to fight cancer?
Yeah, so we can. It sort of depends on how much you know about the cancer. And the other part is it depends on how easy it is to target the cancer. So cancers usually, by the time that they become cancers, they generate a lot of mutations, some of which are necessary for their survival, and other ones they kind of just picked up along the way. And they vary from person to person.
Couldn't you say, though, you could take, a biopsy of, a cancer and, just a biopsy of, the cells of the person, sequence them, highlight any differences, and then, just have the CRISPR go in and specifically target the differences between the cancer cells and the normal cells?
Yeah, so you can. A lot of times one of the issues is that a sizable chunk of mutations in cancer is breaking genes, and fixing broken genes is the hard thing for CRISPR to do. So it's a little bit difficult to kind of revert them back into the normal state. But I will say for fighting cancer, one of the things that's currently in clinical trials is something called CAR T cells. And CAR T cells are a cancer patient's own T cells, their own immune cells, that have been genetically modified to be better at finding and fighting the tumor. Okay. So they're made to be more specific to the tumor. And for that, there's a trial where they've used CRISPR to genetically modify those T cells. I think what you're talking about, I think hypothetically should work. It would be a really serious expansion of what's called personalized medicine because, doing a full genomic read
it
of a
would
person
it would
is
take a lot
expensive.
of resources, especially, in the beginning, but if it's life or death, I mean.
And hell, we're all going to be rich, so it'll help us.
Well, another hard part is still delivery, right? CRISPR is still hard to deliver to any specific one place you want. Mm-hmm. Being sure that you're getting a good amount of CRISPR to tumors, which sometimes are notoriously hard to get things that kill them to, that might be a hard thing, but there's definitely people looking into it. So I think that's definitely something we're working on.
All right, so, we also got... There's all sorts of ways CRISPRs can help with drug screening. There's also ways that CRISPR can help with hemophilia B. Hey, but fuck that shit, okay? We gotta talk about all the sexy sci-fi shit and evil ethical implications of CRISPRs as it becomes democratic, as it becomes terrorist, as Putin gets his hands on it. So let's take a break, and then after that, we're gonna get real fucking geopolitical with CRISPR-Cas9.
Hi guys, this is Andrei. I live near a brewery called Monkish, and it's really good. This isn't a paid ad at all, I just like them a lot. There's a lot of cool dudes there, and they make great beer. There's a cafe nearby too, Fantastics. You can get a breakfast burrito, enjoy it with your IPA. That's pretty cool. You can even bring your dog. I do. He's a poodle named Judas, you'll probably see us there. Again, this isn't a paid ad, I just really like Monkish. Just come and drink some beer. See you there.
Hey guys, this is Petri Dish. We're back, and now we're going to talk about all the crazy, gnarly, ethical, future shit. We're going to talk about biohackers, Osama bin Laden.
You're just doing all the naughty stuff. You gotta calm down.
I don't know if you guys know this, but this is the second time we've tried this.
We had to stop you already.
I get too hot too fast.
Yeah.
But boys, we're going to talk about all the kind of cool ways that we might be using genetic modification, as well as all the ways that it'll inevitably destroy civilization. So Sean, tell us, why would you defend a technology that could be easily used by disparate non-state actors to destroy civilization?
Well, man, CRISPR's already here, dude. CRISPR's already here, and the fact that it's going to get cheaper, I don't think that there's any way that we can turn back the clock on this technology.
Hey, that's what people say about the telegram. Do you see the telegram anymore? Okay.
What the shit does that mean? What the fuck are you talking about?
I mean, the
They
Telegram,
didn't ban
the Telegram is the internet now.
Andre, help me out here. But here, let's start with some of the fun stuff, man. Like, what are some future applications of CRISPRs that you're excited for?
Basically, I think there's a lot of speculative ways that we could do stuff that's either, therapeutic or even non-therapeutic applications of CRISPR. I don't know if you guys know this, but most primates have just one gene broken in the pathway to make our own vitamin C. So vitamin C is a vitamin because we have to get it from our diet, but actually most mammals make their own vitamin C. It's just in primates, we have one gene in this whole long pathway to make vitamin C that's broken, close to the last step. So we just barely don't make vitamin C. I think it'd be interesting to see what happens in people if you just fixed that gene, which you could do with CRISPR, hypothetically.
Okay, that's super boring, man.
No one would ever get a cold again.
Yeah, dude, think about that. You don't have to take airborne no mas.
I love emergency. It just tastes good.
Okay, but another one's like myostatin. You know what myostatin is?
Myoblastas.
All right, calm down. Myoblasts are the correct kind of thing. Those are muscle cells. Myostatin is a protein that inhibits muscle cell growth. So every once in a while, there'll be a person or an animal that has this myostatin gene already broken, and those dudes get ripped to shreds. They get like super muscly. So you'll see some cows that are just like really weirdly ripped.
I think I've seen some pit bulls like that.
Yeah, dude. Yeah, exactly. And those guys are all, they all have a mutation in their myostatin gene that breaks it, and CRISPR's really good at breaking shit. So hypothetically, you could break this gene and then get swole.
Some more swole chads out there.
Exactly.
That's my dream.
Myostatin is actually the gene that one biohacker was targeting when he injected himself in his muscles. But aside from the fact that there's no guarantee that he actually got CRISPR into his cells, even if he did, people aren't sure if myostatin matters when you're an adult. We might need to do it to kids and then make the kids swole. I'm just saying. Some muscly kids.
Okay, what else can we do?
There's a bunch of retroviruses and lentiviruses that have kind of left their shit in our genomes, including ones that are active, right? So some people have latent HIV. Some people have latent herpes viruses or hepatitis B or C. And you can actually break those viruses while they're still latent. You don't have to wait for them to cause the disease. You can break them in the cells that they're hiding in.
Okay. We can get rid of herpes viruses, HPVs, Sep C, Sep B, HIVs.
So what you're saying, though, is because right now, a lot of viruses, once you have them, that's it. Even if they go dormant in your body, they're there still. But there could be a way, in the same way that we have antibiotics that cure bacterial infections, we could actually cure viral infections potentially.
Yeah, so there's a kind of herpes virus called Epstein-Barr virus.
Epstein-Barr!
Which is a particularly topical name now. I don't know how
William Barr, Jeffrey Epstein, had sex, made a baby, turned it into a virus. It's infected 90% of the world.
Yeah,
That's why they silenced him.
In the United States, if you are 25 years old, there is a 90% chance that you have Epstein-Barr virus. And in some people, Epstein-Barr virus causes mono. And in other people, they don't notice that they have it. Some people are infected as kids, and it causes a disease that's kind of similar to a cold for a little bit and then goes away. But a lot of people have this. A vast majority of humanity has this. And rarely, in some cases, it seems to be able to increase the incidence of a lot of other diseases, autoimmune disorders, and cancer. We might be able to break this virus as it hides in our genome, and that would be applicable to most of humanity. And it might be able to save some people from other diseases later.
I mean, Epstein-Barr virus kind of gets us to an important thing, which is what happens when bad actors, right, like William Barr, have the ability to invent viruses that they can, gene hack into you.
Well, so luckily, inventing viruses is not the easiest thing to do. But also, viruses are a little bit overkill for CRISPR, so we've been able to modify viruses pretty successfully for a pretty long time. Luckily, we have not had any bad actors. I don't know if you guys remember this. A few years ago, maybe 4 or 5 years ago now, there was some stuff out in the news about a publishing team of scientists that put out a paper, and then there was basically an embargo on their paper because they described how to make influenza spread easier. So the paper was basically a description of how to make things more likely to cause a pandemic. And the reason for doing that what the scientists were trying to show is that it's relatively easy in the wild for this to happen, and that if we start to see certain mutations in flu strains, we should be really careful because those strains might be able to cause pandemics. So they were doing this, as kind of a scientific-y kind of thing to like see what genes cause pandemics and stuff. But the paper is, it's almost like a recipe book for how to make a pandemic virus.
What would it take for like Osama bin Laden to like, well, he's dead. But for like the next Osama.
So they say.
Yeah, so, oh, fuck.
I haven't seen a body.
Epstein-Barr virus, dude. So what does it take for, what the Epstein-Barr virus actually does is it affects your frontal lobe. So you see reality differently than it is.
Just ask your fucking question.
Red pill, dude. Okay. So what's gonna stop? Like, what would it take for the new Osama to get this, fucking recipe book? What do they need? What kind of resources do they need to then use the recipe book to make some crazy shit, make influenza?
Yeah. So I don't think that there's a very easy way for them to get this paper now, but sort of the underlying work that went into this paper was probably 6 or 7 years of an academic lab's work. If you were really focused on it and you had a reasonable amount of money and a relatively good lab, you could recreate what they did in less than a decade. And once you know that information, then it's some extra work trying it out with different viruses to pick one that you like a lot, like Marburg maybe, or Ebola or something like that, and then trying to figure out a way to make those more pandemic-like. So I think it's possible. Luckily, it's kind of hard to find a team of scientists that want to do anything like that, but it's mostly a matter of money and time.
Something that you and I discussed before recording this episode involved the ability to essentially like create viruses from just their genetic sequences. A fear I expressed was the possibility of like smallpox reemerging through some bad actor who had like a readily available genome of the disease, just being able to recreate it in a lab.
Yeah, so what I'll say is this. It's very easy for you to go to a company and have a string of letters for DNA and say, I would like you to make this string of letters. If it's about 20 to 100 letters long, they can do that for pretty cheap. It is extremely, extremely difficult for you to go to a company and be like, here's an entire, even viral genome, which is thousands of letters. Please give me some copies of this thousands of letters. That would take years of working on to do. Because basically what you have to do, we're really good at printing out shorter segments of DNA, but you'd have to print out a bunch of short segments and glue them together without accidentally.
But we're getting better at that too, I'm sure.
We're getting okay at it. I think maybe the potentially easier thing would be to take a virus that's related, like chickenpox or something, and then look at all the places that chickenpox is different from smallpox, decide on which ones are the ones that like we particularly want to save.
Use CRISPR to make it more like the virus that you want to use to end the world.
Or even arguably, the easiest thing to do is to fly a plane into fucking the World Trade Center, right? No, but being serious though, like, that's why terrorism usually has different forms of political violence than biological weapons.
I think there's a lot of lower hanging fruit. Which, okay, so like for example, I think bioterrorism is somewhat similar to nuclear terrorism in that technically there is fissile material out there, right? I mean, Russia didn't keep a super good track on all of their fissile material after the Soviet Union went down and everything.
Chernobyl.
Iran is doing stuff, North Korea is doing stuff. Hypothetically, a terrorist group could certainly attempt to get this shit together. Now, that said, intelligence organizations are keeping track of that a lot better than maybe they're keeping track of like people buying CRISPR materials in different places. But to me, the question of scientific feasibility is, yes, it's feasible. It would take an academic lab a decade, 20 years of trying. A lot of terrorists don't want to wait that long. I think that's maybe the only saving grace because price-wise, it's going to get cheaper and cheaper. We'll be talking about a couple $1,000, $10,000, well within the means. But time and patience, it requires a lot of training. I think those are the elements that are kind of the limiting factors.
Well, and also political violence. The part of the purpose of terrorism or political violence is to make a clear mission statement, is to project force. And if you just release a disease, then it's a lot harder to successfully project a political agenda, right? It's like, whereas like you fly a plane into the World Trade Center, that's like, you're not just killing people, you're saying, we are this organization, this is our purpose, we exist as a meaningful actor, right? If just like all of a sudden drug-resistant tuberculosis spread more and more in Africa, and it was actually a non-state actor that was spreading it, it would be hard to even know that, right? It would be hard to disaggregate the non-state actor from just like drug-resistant tuberculosis spreading, and it would negate the value of terrorism as a political act.
Well, I hope so. As a scientist, always when I'm thinking about these tools, I'm always thinking about what we can do to help.
I don't buy that. I know you. You're thinking about how we can kill people.
No, I will amend that though. How to help make Pokemon. How to help make Pokemon. That's my main goal. But, I think there's an entire realm of philosophy about bioethics. And I think that bioethics and policy are way behind our scientific achievement. And I don't think that it's my place as a scientist to tell people what's ethical, but I do think it's the place of policymakers and ethicists to fucking talk about it and to set rules and to, talk to scientists about what's possible and then figure out what society wants. And I do think that that's an important step.
But if society puts restrictions on these scientists based on their ethics, can't sufficiently motivated scientists just move their research to an area that is less stringent?
I mean, that happens some, right? It happens with stem cell therapeutics, right?
Yeah. There's
There's people who go to different countries to get like stem cells injected in their spines and stuff, and then they get cancer or something like that. It's that kind of stuff happens, and I don't agree with all restrictions, but I do think that, especially things with gene editing, we've gotten pretty good at it very fast, in like a decade. The number of species that we've been able to genetically modify that previously were just too difficult, it's just skyrocketed. And I think that policy is way far behind on like some more controversial stuff. Like there's this gene that's called monoamine oxidase A. It's in your brains and it's involved in the metabolism of neurotransmitters. Okay, so it can affect behavior. And something that they found is that there's this strong correlation between broken copies of that gene and violent behavior. That there's some people that seem to be more compelled to violence, and a lot of those people statistically have a tendency to have a broken copy of this gene. Now,
If you find the gene, you abort the fetus. I see where you're going.
or what if somebody gets convicted of a violent crime, it turns out that they have a broken form of this gene.
And that's their defense.
That's their defense, or maybe we force genetic engineering upon them. We say, okay, fine, you don't have to go to jail, but we have to get to genetically modify you to no longer have a broken form of that.
I like it. Ice pick lobotomy.
So that's some tricky shit. You know what I mean? And I don't know where to go with that as a scientist. Like that's society's work. That's up to Trump.
You just make it possible, and then you watch society flail around with your discovery.
Exactly.
I feel like you're censoring yourself because I know what you would do.
What would I do?
You would become emperor, dictator, king, and just genetically modify everybody so they have purple hair and no propensity for violence.
I mean, that sounds like fun, but I don't know if I'd do that in like a heartbeat. I think there are other interesting implications. Like, for example, when it comes to disabilities, right? There's a concept of disabilities between what's called immutable and mutable disabilities. So like disabilities you can do something about versus disabilities you can't. And there's sort of like two different classes of stuff. And disability has all kinds of legal implications, societal implications. And for example, some people who are unable to hear or unable to see might not consider themselves to be disabled. They might consider even that concept pejorative to their existence, right?
Sure. Like there's people who could maybe be able to hear with a cochlear implant, but it's like, no, this is who I am. I don't want that shit.
Right.
And
so,
in Sean Science America, we would just force them to get genetically
not
modified.
force them, but if it was cheap and easily available and safe, would people do it to their kids?
Right.
How would that make adult people who can't hear feel that like people are treating the inability to hear as a disease that like they feel compelled to fix. So I'm just saying that there's these ethical implications that are like way bigger than just the science questions.
Isn't that kind of the funny loophole about a democratic society? Is like there's all these rights that we prescribe people, but it's like, but they're kids though. like fuck them.
Yeah, for sure. I mean, I was on the school newspaper. We got stomped on by the principal. Yeah. And then besides all of this shit, like we were saying before, what about putting THC in chicken or something like that?
Yeah.
That sounds like fun.
That would totally be a huge, like, 2-month sale at KFC.
Sounds like something I'm going to do later in my
yeah. I'm just going to biohack some chicken.
Yeah. Injecting THC in our chicken nuggets.
I mean, and you know, I see in the notes that there's, like, 700 gene variants for height. There's a lot of polygenetic qualities to their skin, eye color, stuff like that. Maybe we could start modifying people so that they're all Caucasian.
Are you advocating for this?
You are.
No! I'm advocating for everyone to have purple skin.
Right, you could really get weird with it.
You know?
You should be able to select your hue.
Yeah.
Or the hue of your child.
Right, and you know...
Jesus, guys.
there's all kinds of questions then about like once we start feeling like it's acceptable to have non-therapeutic genetic interventions to like, maybe make somebody, engineer your child so that their metabolism is better. Is it ethically bad to not pursue those? Right? Like if you have the option as a parent to genetically modify your kid to make them have a better life.
Aren't you kind of a douche for being like, actually, no, I want them to suffer.
Right.
I want them to have brown
eyes. There's the potential to have like tiered society.
What's going to happen when we're competing with a bunch of genetically modified individuals for jobs and stuff?
Yeah, this happened in Star Trek.
Like we're here, we're like 70 years old. Like retirement is like now like 130. All right.
Yep.
And now there's these like 20-year-olds that can like lift twice as much.
Yep.
Run twice as fast. Think twice as well. And what are we gonna do?
Yeah, we'll send those kids to outer space.
That's true. Martian children.
Yeah, so they can colonize other planets. Let's make them be able to survive better in low-G environments and shit and just be like, get out of here, buddy.
I think what we've established is we're not necessarily the three people ready to wrestle with these ethical questions well.
Yes, but this shit is coming. And so what I do think is that people should have this on their minds, right? Like we're fucking citizens, you know what I mean? And at some point or another, these issues are going to come up societally, and people need to like at least fucking have some idea that the question exists, so that they can give a little bit of thought on where they stand on it.
That's why you need to share this pod with friends. Share this pod. Nothing we said was offensive.
It's the only hope for humanity.
Elon Musk talking about, Andrew Yang talking about automation. No, dude, genetic modification.
Yeah, sure, yeah. You know what? That's a good segue to wrapping this episode up. I think we did it. So if you want to yell at us over, all the terrible things Nathan said, you can do that on Twitter @DishPodcast. You can email us at petridishpod@gmail.com, and you can donate money to us. I don't know why you'd do that if you're mad at Nathan, but you could donate money to us at patreon.com/petridish.
Special thanks to Stacy Song, our sound lord, to Brian Allen for all the key art, for Andrei Day for joining us.
Yeah,
thank you for having me.
Woo! And yeah, thank you, listeners, for sticking around for our 2-part on CRISPR.
You guys, have a good day.